Okay, this topic is about heredity. In the previous two topics, we already learned about what is DNA and what is a gene. And what is the gene? The gene is actually, the nature of gene is the DNA sequence. A DNA sequence which can be expressed into a product. And this product, in most cases, are proteins. And the thing is, the same type of proteins in a population, or even sometimes in the same individual, is there only one type of a gene? And the answer is no. And there are many different types for the same protein. There are tiny different tiny difference between this version and that version, and but all of those encoding the same gene. For example, the let me see. The one example is the sickle cell anemia. This is the hemoglobin beta unit of hemoglobin, and there's two types: wild type and a sickle cell thing. The two versions, and they only have one difference in amino acids, showing a different shape of the proteins. So in these cases, there are two alleles. And there's another example of this, not a real example, because, for example, the eye color. So just assume there's one gene encoding the eye color. So different people have different genes. And then you have a blue eye, there's the blue gene. A blue gene determines blue eye, and the green gene determines a green eye, etc., and black eye, etc. All different versions, and all different versions of the same gene called allele. And this is not a good example because eye color is more complicated. It's not really determined by only one gene. So, and then accordingly, since we talk about eye color, the eye color itself has another special name from say the setting of genetics. We call eye color it a trait. Trait is a collection, a category of all different versions of the same property. For example, eye color is a trait, and say skin color is another trait. And the trait here is neutral. It does not give you. It only tells you the category. It does not give you the specific description of how it turned out to be. So let's say black eye. That is how a trait turned out to be, and this is another name called a phenotype. And then skin color called a black skin is another phenotype. And what determines the phenotype? One thing determines phenotype is the alleles combination. For each gene in the human body, we have two alleles. So, and this combination of these two alleles, we give another name called genotype. And genotype affects the phenotype, but does it completely determine the phenotype? The answer is no. Two things collectively determine the phenotype. One is genotype, that is environmental factors. So in almost every, not all, but almost every phenotype in human this is the combined effect of genotype and the environment. So this is some of the fundamental basic. Now let's, after learning this fundamental concepts, now let's talk about how genetics work. So let's say there's a long time. I mean, people have observed the phenotype in heredity for a long time. What is heredity? Basically, for example, a kid born to a human is always a human. It cannot be a horse. I mean, a kid born to human parents, she or he always inherited the major features of a human. So this is called heredity. And at the same time, these kids also have slightly different than two parents. And in this is like largely inherited the major features of the parents, but with minor variation. This process we call it heredity. And people have studied heredity for a long time because it's critical for even nowadays. Very critical to improve the, for example, how you can have more wheat from your field. Okay, you increase the agriculture outcome, and make your horses, your dogs less vulnerable to diseases, and all of those may be improved by heredity. And then how? But people has been very thinking about how heredity works, and at the very beginning, this is called blending. A dominant theory called blending heredity. What does it mean, blending heredity? For example, here. For example, if the mother's eye is square and the father's eye is round, and then their kids is a blending of the two shapes, something like this. So this is called blending heredity. And how when things changed, and this is, I mean, nothing changed. So, however, I mean, and then to explain this this blending heredity, people at the beginning believed that there must be something, right? There's something passed from some matter being passed from the mother and the father to the kids, and then this is called a heredity materials, and nowadays we likely call it genetic materials. So at the very beginning, people believe this is a kind of fluid materials. So that's how they kind of call this blending. And until, I mean, actually a way after, there's one of the most famous people in the history of heredity, Mendel. Mendel, who studied the pea plants, and he believed that this genetic material is not a fluid. It's like more like a particle, and each people have two genetic determinant, and then in these cases, these two genetic determinants, like one square, one circle, whatever, and this one cell contain two of those genetic determinants, and when the cell split to produce gametes, each one of those are being passed to one of the cells. So this is the first law of the Mendel's genetics, means the law of segregation. The law of segregation indicating that each person have two genetic determinants, and then this people or this individual generate, not that they have to be people, you generate the next generation, generate the garments. These two genetic determinants are separated to each one of to each of those garments, and so it means these two genetic determinants are separated. That's called law of segregate. And so, for example, you have a father who has two determinants, capital A and lowercase a, the two determinants, two genetic determinants, and mother has another two, also capital A and lowercase a. So how does what his father is generating this? When the father is generating its own gametes, capital A and the lowercase a are separated from each other, and the mother is the same thing. The capital A and the lowercase a are separate from each other, and then the father and the mother they have a chance to combine to each other. They generate all different capabilities, and this is a Punnett square. It's a Punnett square. Please make sure you learn how to make a Punnett square. And the top, actually on the top you list the mothers, and on the side you leave fathers. So basically on the top you separate the two alleles from the mother, here and here, and on the left side you separate the mother's two alleles, capital A and lowercase a, and in the table in between you just combine each of those. In this cell you combine capital A, the two capitals, and in this cell you combine capital A and lowercase a, get this, and get this, and get this. So what's the percentage? And then the kids will inherit it. Both lowercase a is 1 out of 4, which is 25%. That the kids will inherit both lowercase a from both parents. And also this we only talk about the only one alleles. How about there are two alleles or two phenotypes? And that will be the law of independent assortment. Before moving forward, let's say this: the Mendel's heredity is beyond his age, and what is most amazing? I mean, but actually, when nowadays people try to apply his 3 laws of genetics everywhere, in case the reality is that it does not really apply to that many cases. You will talk about this in the future. The law of segregation applies to all the cases. However, the law of independent assortments and the law of dominance does not really apply to every cases. But what makes Mendel's theory really amazing is that actually is outside the 3 laws of inheritance, 3 laws of heredity. Thus, he proposed each individual has 2 independent genetic determinants, which is way beyond his age. So nowadays we know these 2 genetic determinants, that be the 2 alleles, but we are talking about this 200 years after he proposed this. At his age, he does not know what is DNA. People did not know what is DNA at that age, and definitely do not know each person has 2 copies of DNA. So next, let's talk about the law of independent assortment. So what does it mean, the law of independent assortment? For example, let's make a easy example. In my lecture notes, I put a real example of the pea plant's flower color and the shape of the seeds. Let's make some easy to understand example. However, it does not. I mean, just keep in mind, it's only an example. In a real human, it does not apply. Let's say eye color and hair shape. Okay, in the real world, none of those is determined by only one gene, and it's followed the Mendel heredity. But just make an example, and let's say this: there's the father here has let's say the eye color has 2 phenotypes. The black, and now talk about human, then red. Okay, we talk about maybe, I don't know, some hypothetical monster. The black eye, B, 2 capital B, and 1 capital and 1 lowercase b will be black eye. And you may heard of this is called a dominance, and we talk about this law of dominance later. And also we talked about the law of dominance actually only apply to very few phenotypes. But anyway, let's start with this one. So both capital B, capital, if the guy has both a capital allele, this B gene, this guy has a black eye. And if the heterozygous, one capital, one lowercase, also black eye. And the right eye, lowercase b, lowercase b. So this is right eye. Maybe let's change different way. I don't really like this capital lowercase. I don't know who invented this to make it easier for. But let's use a different one. This is B and R. So B and B, two black alleles result in black eyes, and two R alleles result in red eyes. And then the question is, what is BR? If following the law of dominance, we talk about later, BR is black, so R is recessive, and B is dominance. And the hair shape, then there are also two hair shapes, straight and curly. Curly, yes. So S means straight, C means curly, and then SC is straight, means straight is dominance, curly is recessive. So this is two alleles. Two phenotypes. Each phenotype has two alleles. And then let's say the father. I want to say father first. Say mother first. We put the mother on the other side. Mother. Mother's genotype is black eye and straight hair. And the father is let's see. No. So also black eye and straight hair. However, this is the phenotype, and the genotype is none of those are both of those are heterozygous. None of those are homozygous. So in these cases, let's say, let's make a big Punnett square. Here put mother. So B can combine with S. So one of the genotype of the gametes is B and S, and then B can combine with C, that's BC, and then R combined with S, RS, and then R combined with C is RC. So these are four different types of the gametes that this mother can generate, and the same apply to the father. BS, BC, RS, RC. So the question is, do you think this B allele can always associate with S allele? If the B is always combined with S, then the BS has a much higher chance of combine together to generate a BS. So the percentage of BS should be more than BC. So in this case, you can have BBSS. So BBSC, BRSS, BRSC, etc. BBCSC, BBCC. B R S C B R C C B R SS. So you just do all of those combination, and C, SC. Oh, yeah, that make it more difficult to. Yeah, too much. I don't want to write it. Yes, whatever. Okay, we got all of those. But the thing is, if B, the black hair, the black eye, and the straight hair, B and R always, if they linked to each other, then the B and S has B and S percentage is should be BS. The percentage should be way higher than the B and C percentage. In this cases, this B, B, S, S should be way more than B, B, not this. A little bit complicated. We have to calculate this one. Theoretically, you have how many? It should be, let's say, you can do your calculation at home, sorry. Eventually the black and straight versus red straight versus black curly, okay, versus red curly should be 9232323, and this still exist. And actually this showing that, let me cut this calculation. It will take much longer than I thought to explain this calculation, but still this one. So the B and S combined is way more than B and C. If they are B and S are basically they are linked to each other, the BS combination should be the percentage B and S combination should be much higher than BC combination. And the reality, after Mendel's experiment, when he do all of those math and the binding, he shows that no, that's not true. The BS combination is equal to BC combination, which means these two, the eye color and the hair shape, are independent to each other. So this we call independent assortment. So which means for the independent assortment, it means that for the two phenotype, these two phenotypes are being their alleles being distributed to the next generation independent to each other, and there's no strong association between each two of those. That's called the independent assortment. The thing is it true? Is it true? The thing is, it's only true in Mendel has find out 7 different alleles in the pea plant. For example, flower color, the shape of the peas, and the height of height of the plant. But that's only 7 of those. To follow this, all of those are separate from each other. They're all many different things. And when people study more and more phenotypes, they realize it's not always like this. And this is called genetic linkage. So what is genetic linkage here? So there's another guy called Morgan. Morgan is like this? Morgan, maybe. So he studied the fruit fly. In the case of the fruit fly, there's a long antenna and long wings. In comparison, there's also short antenna and short wings. If is follow the law of independent assortment, the long antenna has equal chance to combine with short wings, and the long wings have equal chance to combine with short antenna or short. Sorry. The long antenna has equal chance to combine with the with long wings and short wings, and then the short antenna has equal chance to combine with short wings and long wings and short wings. But in reality, long antenna and long wings are always bind. Has a much higher chance to show up together in free flights, and short antenna and short wings have much higher chance to show up in the free flights. What's the cause of this? That's because, and it's also proposed by Morgan, that Thomas Morgan, that here these two genes, this is the antenna, and this is wing length, antenna length and the wing length. The genes that determine antenna length and wing length are very close to each other in one chromosome. So that's the key thing about here, is that it indicating what's the heredity units here. What's the unit of heredity? Is this one single gene, the unit of heredity? No, it's the chromosome. Chromosome is the units of the heredity. When the parents pass genetic material to the next generation, it actually passed a whole chromosome, not a single gene. So chromosome is the unit of the heredity. However, we're not going to go to details in this way. Even though the alleles in one chromosome, there's still chance they'll be separated by another process called crossing over, which happened during the meiosis 1. And we are not going to details of those, but just keep in mind, even though the genes in one chromosomes, they have a chance to be separated. And the chance that they will be separated, it depends on how close you are. That's why I say the genes that determine the antenna length and the wing length are located very close in one chromosome. So now let's talk about another law of the Mendel's heredity, law of dominance. So this may be the most well-known law of heredity, but it's not. It may only apply to very few phenotypes. Let's go back here. So say black eye. Let's say black and red eye. So allele black, this is black eye. Allele R and R is red eye. And what is allele RBR? According to the law of dominance, it's completely black. So this is dominant, and this is recessive. How many, in the cases, how many of those genes really existing follow exactly this law? The truth is very few, because there's a large limitation of this law. Firstly, there's only two alleles. In the case, if you want to make this law of dominance apply, there's only two alleles, and then only two phenotypes, and then one gene fully determine these phenotypes, that not even environment. So there's so many things you have to, so many conditions you have to meet to be so that the law of dominance can apply. In the real world, rarely this law can be applied. In human, there's only a handful of disease or phenotypes really fits into the law of dominance. In most cases, firstly, the phenotype is quantitative phenotype. For example, body height, body weight, skin color. We talk about black, dark, black and white, or dark color. But the truth is, there's not just a simple black and white. People's skin color has a gradient tone. You look around, right? And we say the hair shape, curly or straight. If you look into the very few people have the fully straight hairs, and many different things, many metabolism, the heart function, many of those are quantitatives. So in most cases, in human, the phenotype is quantitative. And how you can determine this quantitative, how this quantitative phenotype is determined. Actually, now here, it's more fit into the beginning. We talk about the blending heredity. So the blending heredity actually can explain the phenotype. And normally, for example, you have a white person marry a black guy, and then their kids is very likely the skin color, skin tone is in between. But the thing is that the original genetic theory can only tell the phenotype blending, and then it wanted to explain this because of liquid material behind it, which is not true. Okay, there's no. It's true to tell the most of the cases of the phenotype, but in the case of the genotype, it's not true. It cannot explain the genotype, and what kind of genotype can result in a quantitative phenotype. That's because these things, this quantitative phenotype, are determined by, we call it apology. What is apology? Tens, hundreds, thousands. Sorry about my handwriting. Thousands of genes to determine one phenotype. So that's the most common situation in human heredity. Apology determining a quantitative phenotype. That's the most common way. And again, the law of dominance only can apply to a very few of genes in the case of a human, in the case of a human phenotype or diseases. The most common case is the apology determining a quantitative phenotype, and then in between there's some other genetic say phenomenon. Let's briefly discuss this. What we call it incomplete dominant. What is this? Again, we talk about black eye and red eye. Black eye is BB. Red eye is BR. How about, I'm sorry, red eye is RR. And if it's complete dominance, the BR should be black eye. However, in the incomplete dominance, it's brown. It's somewhere between red and black, so it's brown. It's not. So this is called incomplete dominance. This is not a good example because the eye color again is complicated. But the real example is this. Let's talk about sickle cell. Sickle cell disorder, and this gene called sickle cell HBS. So HBA, 2 allele HBA is healthy, and 2 allele HBS is sickle cell. Sorry. What if HBA and HBS, in general, they are healthy? Both. So that's we call it a sickle cell is a recessive disease. Recessive disease. That's the common way we talk about is. However, is not totally true. The heterozygotes HBE and HBS, they are actually vulnerable to hypoxia. They are not completely the same as the healthy people. In oxygen stress, for example, they climb to a high mountain, or in a very intensive exercise, when the body needs tons of oxygen, their hemoglobin may change their shape and become a sickle cell. So that's called incomplete dominance. The heterozygotes are not completely the same as the dominant homozygotes. Another thing is called multiple alleles. And what I mean by multiple alleles, before we talk only B and R, this one. How about you have another say G, it's GG, that's green eye, and then there's more thing you talk about though. Okay, you have BG. What's a BG color? I don't know. I only made it up. And but here we can still using the sickle cell as an example. We talk about HBA is the wild type, the healthy one. HBS, sickle cell. Actually, you have people also have HBC, HBD, HBE, and all of those are different versions, alleles of to determine the same hemoglobin, the gene, the beta units of hemoglobin. And what are those things? What are those stuffs? If they have different combinations, then they're different weight, but then they all show different phenotypes. So we already say HBS, HBS, this is the most severe form of a sickle cell. However, if people have HBA and HBC, it's largely fine. However, if a guy has HBS, one allele of HBS plus HBC, there's still a sickle cell, although it's less severe sickle cell. So this is called multiple alleles. And now you have more than one combination. So make things more complicated, and more than two alleles, multiple alleles. And then one more thing is called co-dominance. What is called co-dominance? And now we have to use example ABO blood type. You know, with A and B and O blood type. A blood type, all of those, this is determined by only one gene. And then double A alleles will be A blood type. A and O is AO blood types. Is also A blood types, and B and BO all B blood types. So A or B are dominant over O. So only OO are O blood type. How about AB though? In the case of AB, the blood type is basically determined by the red blood cells here, and then on surface there are proteins. Okay, A blood type, it have A type of sugar modification, and B blood type, you have a B type of sugar decoration. And then in the case of, if this guy has both A and B alleles, then this guy will display both A type and B type on the surface, and this is an AB blood type. And in this case, both A and B's phenotype are displayed on the surface of cells, and then this we call it co-dominance. No one can hide another one. And this is one last thing. Technically, epistasis. In the case of epistasis, there's two genes. One gene 1. For example, this is most studied in the fur color. In G1 is, say, black and yellow. Black is dominant, yellow is recessive. So BB, BY are black, YY are yellow. However, you also have gene 2. In the case of gene 2, no color or no color. So C, 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 N. C is null over N. NN is no color. In the case of either CC or CN have some color, and this color determined by black or yellow. And when there's NN allele in the gene 2, then there's no color at all. In this case, it doesn't matter. It doesn't matter what's the genotype of a bee. It's always white, albino. So this is epistatus. It means one gene completely override the phenotype, another one. So if one gene turns into a certain phenotype, then there's no any variation of the phenotype. It doesn't matter it doesn't matter whatever the other gene turned out to be. So that's all for the heredity, and thank you all for watching this.